Okay, good morning. We are Group 11, and our topic was on applied microbiology, antimicrobial drug research. And the article we chose was the antimicrobial drug discovery through bacteriophage genomics. Our group consisted of
Kiana Harris.
Terrence Mitchell.
Sydney Sims.
And Jasmine Whitaker.
And here's our presentation.
I am now going to give you an introduction to the article we are presenting and the topics it covers. First, we have the history of antimicrobial drug discovery. Antimicrobials are those drugs that target microorganisms. They do this by destroying or interfering with microbial structures and enzymes. They have revolutionized the treatment and management of infectious diseases, which were once the leading cause of mortality in the human population. Now we are going to discuss bacteriophages and genomics. A bacteriophage is a virus that parasites a cell and reproduces inside of it. They are harmful because they play a role in human disease by turning harmless bacteria into agents of disease. They have unique proteins that attack critical cellular processes. The information in the phage genomes could be extracted and used to identify certain proteins in their bacterial host and to prioritize these targets for drug discovery. Genomics is the branch of molecular biology that is concerned with the structure, function, evolution, and mapping of genomes. Below is an image that shows what a genome of a bacteriophage looks like. It shows the cell functions and where they occur, such as cell lysis, DNA and RNA metabolism, and even the unknown functions. On this slide, you will see the life cycle of a bacteriophage and what they look like up close. They are composed of a head and a tail. The head hosts the protein and DNA, while the tail only hosts the protein. Lastly, I will connect all of this to the article. They used the gram-positive bacterium Staphylococcus aureus and sequenced 26 phages and identified 31 polypeptide families that inhibited growth. The bacterium is an important cause of severe infections in immunocompromised individuals. The completion of the sequence of the bacterium, genome 4, allows the use of functional genomic strategies, which are used to identify essential genes and proteins for new antimicrobial drug development. In the article, they hypothesized that they could use the concept of phage mediation inhibition of bacterial growth to systematically identify antimicrobial phage encoded polypeptides.
All right, so now I will be discussing the materials and methods. So phage genomic DNA was randomly cleaved by sonication, and the ends were repaired with C4 DNA polymers and clonal fragment and cloned into the Pig 11 site of plasmid PKS. Inserts were sequenced using either the Big Bye primer or the Terminator cycle sequencing reaction. So the sequence contents were several using either the sequencer 3.1, also known as the Gen codes, or the FEPFRAP/content 12.0, which is also known as the Codine Code Corporation software. When bacteriophages began at the first nucleotide, which is then scanned for a start codon. So when the bacteriophage is identified, the number of intern codons is counted until termination codon is reached. A minimum of 33 codons define the sequence as ORF, and this procedure is repeated until the end of phage sequence is reached. The scan is performed in an identical manner on all three readings, framed of both DNA strands in order to identify all the fugitive RNA. Fugitive genes are then identified based on the presence of a Cheyenne-Delgarno sequence within the first 50 nucleotide upstream of the start codon. So DNA encoding individual phage ORFs were amplified by the PCR from phage genomic DNA and cloned into the MCS of the modified vector. Phage encoded inhibitory polypeptides were expressed as either GST or His6 fusions and purified from E. coli. Yeast 2 hybrid analysis was performed, and T antigen and lamin C were used as controls for the autoreactivity. 0.91 qB DNA fragments started from the ribosomal binding site were amplified by the PCR from the S arterioles and sequentially cloned and inserted into MCS. The resultant plasmid, which contained two PCR fragments, was then used to transform the S arteriole strain to allow homologous recombination. Small molecule compounds were then screened at 20 micrometers in a 10 micrometer area. Then lastly, proteins were incubated with compounds for 2 hours at the ambient temperature of 22 to 25 degrees Celsius, and the fluorescence polarization was determined in 384 well proxy plates using an ultrafluorescence plate reader called a TCAN.
Okay, so in the results, when monitored the expression of 77-ORF-104 within S. aureus in liquid culture, polypeptide was found in a bacteriostatic effect due to an arrest in cell growth. Small quantities of the strain were expressed, suggesting that the growth did not experience overproduction. 69 predicted ORFs in phase 77 were screened, and only 3 strains were found to inhibit S. aureus. And in the figure to the right, it just shows the growth medium and the production with the help of the enzyme IPTG. And so for A, it shows that the bacteria experience growth with the help of IPTG. And to the right, it did not without the enzyme IPTG. And for figure B, it shows the growth as well, just with the addition of the protein DNAL and DNAD. So by extending the screen to 895 ORFs from the additional 26 sequence phages of S. aureus, 31 distinct families of growth proteins were found. A single polypeptide of 38 kDa was identified as the S. aureus homolog bacillus subtilis DNAL. A series of cell-based and in vitro assays were performed to confirm 77-ORF-104 was associated with DNAL. The results conclude that DNAL is an essential gene of S. aureus. And this figure shows the growth of DNA and RNA. So in A, DNA decreases with time, while RNA also decreases with time, but not as much as DNA. And for figure B, RNA decreases while DNA increases.
So in conclusion, the results from our study showed that by mimicking the growth behaviors, we can form new antibiotics to fight against different infectious diseases. The clinical significance of this is that it allows medical professionals a new access to a way to fight different new mutations of bacterial strands that cause a lot of the infectious diseases that we are known of today. This opens a whole new world, medically speaking, of different treatments and preventatives, such as using more homeopathic medicinal reasons rather than traditional Western medicine. And some examples would be in essential oils, different genomes of combats, just for gene sequencing and stuff such as that. These are references. And we'd like to acknowledge our microbiology professor, Dr. Shrita Cooks-Fagwellin, and also the researchers and writers of this article for putting their time and effort into just coming up with the new form of medicinal treatments for different patients.
We would like to thank you for listening to our presentation, and we hope that we were able to teach you about the antimicrobial drug discovery through bacteriophage genomics. Thank you.